86 billion neurons. That's roughly what's inside your brain right now, continuously firing and forming trillions of extraordinarily precise synaptic connections every single second. Welcome to this explainer on advanced neural architecture and physiological mechanisms. If you're studying the biological sciences, you already know the staggering complexity of the nervous system, but today we're moving entirely beyond the basics. We are going to map exactly how microscopic cellular structure dictates complex physiological function, examining the nervous system with exacting anatomical precision. Let's dive in.
Here's our roadmap for the explainer today. We'll be going through 1. Nervous tissue architecture, 2. The anatomy of neurons, 3. Glial cell support networks, 4. Action potentials and myelination, and finally, 5. Synaptic communication mechanisms.
Section 1. Nervous tissue architecture. To start, there's a really stark, fundamental structural dichotomy between the central nervous system and the peripheral nervous system. The CNS, so your brain and spinal cord, operates as a heavily fortified, highly compact matrix. It's totally dedicated to dense central processing and integration. By contrast, the peripheral nerve bundles of the PNS stretch way outward to connect that central processing hub to the body's furthest extremities. Now, these bundles house both somatic and autonomic fibers, and to ensure rapid, continuous signal transmission across those vast physiological distances, some individual peripheral axons within these bundles can literally stretch over a meter long. We're talking about a single, continuous cell running from the base of the spine all the way down to a single toe.
Section 2. The anatomy of neurons.
So zooming in on those primary communicators, the neuron itself exhibits profound functional polarity. It's essentially a one-way street. Electrical signals arrive at the highly branched dendrites, which gather inputs from thousands of adjacent cells. All of that electrochemical information then funnels directly into the soma, the central cell body housing the nucleus. Here's where it gets interesting. All of these incoming signals, both excitatory and inhibitory, are spatially and temporally summated at a specialized region called the axon hillock. If the accumulated membrane potential there reaches a critical threshold, it triggers an all-or-nothing action potential that propagates down the elongated axon toward the next target.
Section 3. Glial cell support networks. But neurons absolutely cannot survive or function on their own. They rely entirely on a robust cellular support cast known as glia. Let's look at 3 major players. First, astrocytes. These are highly versatile, star-shaped cells in the CNS that do far more than just offer structural scaffolding. They actively regulate extracellular potassium levels and tight junctions to maintain the highly selective blood-brain barrier. Next, oligodendrocytes, which are responsible for generating and wrapping the vital myelin sheath around CNS axons. And finally, microglia. Uniquely derived from embryonic macrophages rather than neuroectoderm, microglia are constantly patrolling the CNS parenchyma. They actively phagocytose cellular debris and mediate the brain's innate immune and inflammatory responses.
Section 4. Action potentials and myelination. Now, let's talk about the mechanism of an action potential, because it's a true marvel of cellular biophysics. It starts with a rapid depolarization phase, driven by the opening of voltage-gated sodium channels, which allows a massive, localized influx of sodium ions right into the cell. This is rapidly followed by repolarization, governed by the subsequent opening of delayed rectifier potassium channels that allow potassium efflux. As this cycle completes, the action potential propagates distally along the axon. But in highly evolved nervous systems, this signal undergoes what we call saltatory conduction, completely bypassing continuous propagation for a much more efficient method of travel.
To understand that speed, we have to look at the myelin sheath. It acts as a high-resistance, low-capacitance electrical insulator. But it isn't perfectly continuous. It's periodically interrupted by microscopic gaps known as the nodes of Ranvier. At these highly specialized nodes, the axolemma is densely packed with voltage-gated ion channels. Because the myelin prevents ion leakage everywhere else, the action potential is effectively and completely regenerated at each successive node. So the electrical impulse literally leaps rapidly down the axon. This specific mechanism allows signals to travel at staggering speeds of up to 120 meters per second. Compare that to a sluggish 1 to 2 meters per second in unmyelinated fibers, and you see the difference.
So, myelination is a massive evolutionary advantage, but it actually goes way beyond sheer signal velocity.
It exponentially increases the metabolic efficiency of the entire neuron because the cell only needs to actively expend ATP to run its sodium-potassium pumps at those discrete nodes of Ranvier rather than along the entire continuous length of the axon. It conserves a tremendous amount of cellular energy. Simultaneously, that same myelin sheath is providing robust physical shielding and trophic support to the delicate axoplasm housed inside.
Section 5. Synaptic communication mechanisms. Eventually, at the distal terminal of the axon, that electrical journey hits a definitive physical roadblock, the synaptic cleft. We're talking about an ultra-narrow extracellular space, typically measuring a mere 20 to 40 nanometers wide, that separates the presynaptic sender from the postsynaptic receiver. Because an electrical current simply cannot bridge this fluid-filled gap on its own, the advancing signal has to undergo a fundamental transduction. It has to change its medium entirely from electrical to chemical to successfully reach the target cell. So how does that happen? When the action potential invades the presynaptic terminal, the depolarization triggers the opening of voltage-gated calcium channels. The resulting rapid influx of calcium ions mediates a completely critical process. It causes synaptic vesicles to dock and fuse with the presynaptic membrane via snare proteins. This mechanism of exocytosis floods the synaptic cleft with specific neurotransmitter molecules. These chemical messengers then rapidly diffuse across that tiny 20 to 40 nanometer space and bind right to specific receptors on the postsynaptic cell, inducing immediate ligand-gated conformational changes.
This precise chemical transduction represents the ultimate functional outcome of all the advanced neural architecture we've explored today. It perfectly translates fleeting, microscopic electrical impulses into sustained, highly complex physiological actions. Whether it triggers an excitatory postsynaptic potential in an adjacent neuron, commands a striated muscle fiber to contract, or signals a neuroendocrine gland to secrete its hormones, this microscopic molecular handover is the exact foundation of all synchronized biological harmony in the body.
Grasping this flawless baseline architecture is absolutely crucial. It's the required first step before we can understand the devastating realities of neuropathology. Because if this exquisite machinery ensures perfect macro-to-micro communication, we have to ask, what happens to the human body when it deteriorates? Think about demyelinating conditions like multiple sclerosis, where an autoimmune attack on oligodendrocytes progressively strips axons of their protective myelin. The resulting conduction block drastically alters macroscopic motor and sensory functions, which powerfully demonstrates just how deeply human vitality relies on microscopic structural integrity. It's a heavy but fascinating reality to consider as you continue your rigorous exploration of the biological sciences. Keep questioning these mechanisms, and thank you for joining us for this explainer today.